minutes. So let's try to finish 30-35 minutes and 10 to 15 minutes for interaction. Okay. So is it moving? So again stuck. So all these times should not count in my presentation. Or if that keeps working, that's also fine. If you can put up these lights, I think everything works. Thank you. We are troubling you so much. So moving on to, so I think the simple way to get yourself into this presentation is, you all might have heard about Rip and Winkle Uncle, right? We used to study about this story. One who went to the forest and he slept for a long time, 30, 40 years, and then he came back. Suppose he comes back to a city like New York, right? He has seen only a small village 30, 40 years ago, and now he is in New York among the skyscraper buildings. How he will feel? Similarly, a plant breeder of 1930s, 1940s, even 1950s, 1960s, let's assume he goes into a deep sleep. And then suddenly in 2019 or on the onset of 2020, this guy comes back and then sees what's going on right now in a modern plant breeding program. So if that is the context, what are all the paradigm changes he will see? What he was doing, say 40, 50 years ago, and 40, 50 years after, what are the changes that he is seeing and how he is interpreting all those changes. So let's assume that way and move forward. So the first paradigm shift, I'm going to talk about 5 paradigm changes. The first one that I'm going to talk about is line development. So there have been significant changes with respect to line development. So as you might have understood, in any hybrid crops, there are 2 phases of work, right? So one is line development, the other one is hybrid making and evaluation. So any breeding program spends 50% of the resources roughly on developing lines, and then remaining 50% on making hybrid combinations and evaluating them in the field. So any given hybrid in a cross-pollinated crop has 2 parents, right? So you have parent 1 and parent 2 if it's a single cross hybrid. If it's a 3-way cross hybrid, it will have more than 2 parents. But so as you would have understood, the parental lines are really the foundation of hybrid breeding. So how do we develop these lines? It has gone through a significant change in the past at least 2-3 decades. So how many of you have heard about double haploids? Many of you have heard about double haploids. Very good. So double haploids is one of the significant change in corn breeding that is kind of revolutionizing line development. In the past, how we were developing inbred lines in corn? We were making breeding crosses and we were passing them through multiple filial generations, like 6 to 7 generations in a year, over a period of 4 to 5 years, and then you will end up with a homozygous line, which is called an inbred line, right? So this used to take at least 4 to 5 years, whereas now what do we have? Double haploid technology. There is a special inducer. So there is a special genotype in corn. It's called inducer genotype. Corn is very fortunate to have this, naturally existing. So when you cross a plant to this inducer pollen, so it gives out pollen, but then that pollen doesn't participate in the fertilization process. It's like, a wedding is happening, but then kids are born solely out of mother's genome. So there is no paternal genome at all. So that's how the haploids are formed, and the haploids are doubled, you get double haploids. So this is a technology that is really changed the phase of corn breeding, other than combines and molecular markers. So combines and molecular markers are other 2 technologies that have had greatest effect. So this is the process that I described earlier. So there is a manual published by CIMMYT, the International Center for Wheat and Maize Improvement, and they have very clearly documented all the processes and what are the different applications possible in double haploids. So I would urge you all to go through that document and learn more about double haploids. Okay. In fact, annual output of some of the breeding programs, millions of double haploids per year. In the past, we were developing 500 to 1000 inbred lines when we were doing the conventional process. Because of double haploids, now we are generating millions of double haploids in the breeding program. So that helps increase the genetic gain. Breeding still continues to be numbers game. The more you generate, the more probability of identifying a good genotype. So that's a technology in corn. But not all crops are fortunate to have that kind of inducer genotypes. In other crops, what's happening? How about developing haploids through male gametes? So what we did in corn, what I described earlier, was developing progenies from only the female line. Whereas this one, can you generate the pollen? Can pollen be a donor for haploids? So can we generate haploids out of the microspores? So this technology is again very, very well optimized in mustard and canola. So just seed to seed is just 8 months and you have fixed lines. You can generate as many DH lines as possible. Otherwise, when you do it conventionally, it takes at least 7 to 8 generations over 4 to 5 years period of time. Whereas this is squarely within 8 months you are developing the fixed lines. So that's another important development. How about crops wherein neither microspore works nor double haploid works? What else can we do? Have you heard about rapid generation advancement? So it is nothing but what you would have studied as a plant breeding student or a molecular breeding student, SSD, single seed descent. It is a way of developing mapping populations. So just similar to that, it is nothing different from the conventional plant breeding. Everything is same. You are passing the population through a series of multiple filial generations, but then you are doing this in containment structures. So instead of one season or 2 seasons at the most in a year in the field, here you are able to raise them in the greenhouse conditions at least 4 generations in a year. So significant advancement as against the conventional method. So this is again revolutionizing. And can you give me an example of where it is used? It's there in the title of the slide itself. So it is used in rice. So Erie as champion pioneered a lot of developments in this. At Corteva, we use rapid generation advancement quite a bit, and this is resulting in large number of fixed lines within a very short period of time. So that is the first paradigm that I wanted to talk to you. So 40 years ago, none of this was available to any of the conventional plant breeders, right? Whereas now all these technologies are available. And in fact, 95% of the breeding program uses these kind of technologies. Okay. So the next paradigm shift that I want to talk about is selection based on genotypes rather than phenotypes. So there is a very popular saying that seeing is believing, right? Seeing is believing. There is a popular saying in English. Seeing is believing is not true with respect to plant breeding. What you see can be really misleading. So when you go to a location, what you see, a genotype producing the biggest corn ears need not be the same in another location. So that is a traditional G by E effect. So what you see in one location is not representative of what you see in another location, right? So that's why it's really important to understand, to go beneath what you see in terms of phenotype and understand the true genetic value of an individual. So the true genetic value of an individual can be understood by studying its genotypes rather than phenotypes. So that's a significant change which is happening in recent days. So why we are talking so much about marker assisted selection, molecular breeding, everything is because phenotyping is getting really, really expensive. Going and measuring phenotypes in the field is really expensive because land is very expensive. Manpower is expensive. Whereas if you want to generate millions of data points, especially after the onset of SNPs, it's really, really cheap. So your data cost per data point for a genotype versus cost per data point versus phenotype, there is a significant difference, and genotype is low cost, whereas phenotype is becoming high cost. And also many a time when you wanted to measure phenotypes like drought, it suddenly rains and then you lose the experiment, right? So all this it's becoming very uncertain, and that's why we are talking about other methods of selection, genotype based selection. When I was a PhD student, we were using mostly RAPD markers. Fortunately, I was not there during the RFLP time, so many of you might have worked on RFLPs, such a difficult job. In my time, RAPDs came and then SSRs became really popular, right? Whereas that time to run 200 markers, I think my whole PhD work would have been like that, right? Whereas now, after the onset of SNP markers, 200 SNPs, 200 markers, if you ask Karun, I think our production genotyping manager, he will do it within I mean less than an hour or so, he will finish the job. What we did in 3 years, I think the current labs can do it in less than an hour. So that is kind of creating a revolution, genomic revolution, and we are adding a lot of data points, but the real challenge here is you are generating millions of data points, but how are we making sense of those data points in a breeding program to develop varieties? So that is the real challenge, and that's where next generation plant scientists like you will play a very significant role. So if you ask me broadly, all the genotype data that we are generating, where is it being used? You are generating high density genotypes on one hand, high precision phenotypes on the other hand, you have powerful statistics, high efficiency computing, all that. So it is all getting used in two big streams. One is mapping a trait. There are certain clearly factors, genes responsible for some of the qualitative traits, so you can map them, identify markers, and use that in the breeding program. That is one way of using it. But then not all the traits are amenable for that approach. Say for instance, if you ask for a molecular marker for grain yield, does anyone have molecular markers for grain yield? No, you don't have 1 or 2 markers associated with complex traits like grain yield. Whereas for quality traits, for simple traits, we do have clearly markers. So what about complex traits for which, how we can use molecular markers? So that's where the new approach called genomic selection has come in. How many of you have heard about genomic selection? I'm good. So there are so many, I'm feeling glad so many people have heard about genomic selection. So genomic selection is nothing but you are trying to make a prediction of a line without actually testing it in the field. So without extensive field evaluations, can we understand the true genetic value of a line just by making use of genotypes? Yes, it is possible, and this technology has evolved quite a bit in the last few years. So we will have a small demo tomorrow on genomic selection. So this is the thing that I talked about. I'm sure many of you might be very familiar with these traits that I have mentioned: BLB, rice blast, BPH. All these traits have simpler genetics. There are 1 or 2 large effect loci controlling most of the phenotypic variation. It's easy to identify, use it in the breeding program. If it is there, yes, you are good. Go ahead and use it. But then unfortunately, many of the traits are not like that. Many of the traits that the breeder is interested, the grain yield for drought tolerance, nitrogen tolerance, all these low nitrogen tolerance, all these traits are very complex, and you need a different approach, which I explained to you, which is genomic selection. So you have a training population which is characterized phenotypically as well as genotypically, and then you estimate marker effects based on this. And then when you are given a new set of line which have not been tested until now in the field, you have an opportunity to understand the true value of that individual using the training set that you have. So this is just a primer. Go back and study on these latest developments. Genomic selection, very interesting. So in terms of the simple, in terms of how to understand this is, you have a population of individuals. You are just sampling few individuals from that population, not every individual. You are sampling few individuals from that population. You are characterizing it genotypically as well as phenotypically, and then using that you estimate some marker effects. Use that marker effects to evaluate a new upcoming line in your breeding program. So one of the prerequisites for this kind of genomic selection to work, we also go and talk to many of the public sector breeding programs. Everybody is excited about using this, but this is not as simple as we would use in the other approach, which is like identifying 1 or 2 genes, tagging with molecular markers, and using that marker in the breeding program. So this is not that simple, right? So here what you need to do, your breeding program has to be basically very well organized. Only then you will be able to make use of genomic selection approach. So what do I mean by that? So if you see these dots are nothing but all the inbred lines in our company, Corteva. Each dot represents an inbred line in the corn breeding program. Okay, so when you see like this, everything looks like very unrelated to each other. But then what has happened in the company, one of the strongest factor is that in the last 100 years, whoever has done breeding, whatever he has done, everything is documented very well in the informatics pipeline. So how a founder population 40 years ago relates to a current inbred line is very, very well documented. So if you see now, it's not playing. So it's a movie, so it would have played. Side screen is playing. Okay. So what you see here. So what you see here. Okay. What you see here is two heterotic groups. Sorry. So what you see here are two heterotic groups. One is a male heterotic group. The other one is the female heterotic group, which we call NSS and the SS. And these are two groups that predominantly used in the temperate corn breeding program in the US. So it's important to organize the germplasm into heterotic groups to be able to apply genomic selection later in your program. So I was just stressing upon you that it's important to keep the breeding program information documented and organized to be able to use the genomic selection in your approach. So that's one of the important prerequisites. So with all these DH molecular markers, what has happened, this is an important slide that I want you to understand. What has happened is, have you heard about the breeding funnel, the famous breeding funnel? So this is basically nothing but, you see this funnel. So it's so wide, right? So that means too many recombinants are going into the funnel, and through multiple evaluations over the environment, you are releasing a handful of commercial. So 10,000 double haploids are going in and few hybrids are getting released. So the funnel is wider, the funnel is shorter. It is because of the developments now, whereas in the past, so the other one is also not showing. In the past, what used to happen is the funnel used to be very narrow, small, only a limited number that you can pour in, and it used to be very long. So it was taking more time for you to develop variety, whereas now it is taking considerably less time because of developments in markers and the genotypes. Okay, so that is a paradigm shift with respect to molecular markers and selection based on genotypes. So third paradigm shift that we are seeing is selection based on the phenotypes is still there, but then phenotyping is getting very, very digital, very precise, high throughput, and statistically intensive. So just observing on a notebook, on a random replication of 3 randomized replication, 3 will not do, and taking an average will not do. Why? So these are some of our old approaches of measuring traits, whereas of late we have developed combines, we have developed customized machineries for measuring plant characteristics. Now, one simple thing, how many of you have done QTL mapping for your MSc or PhD projects? QTL mapping? Yeah, so many of you have done QTL mapping. So after you generate the phenotype, have you been able to correct those phenotypes for the experimental errors in the field? Now, one thing given in agricultural research is whatever field experiment you will do, you will invariably have error component. There will be an error in agricultural experiment. So it is important to recognize that error and correct for that error. So if you see in this photograph from left to right, so there are one row which is kind of stunted. It is because of the overwatering. In another field, there may be water flooded in many rows. And then in another thing, the edge effect, the plants on the border, they are bigger than the plants inside the plot. So all these are nothing but errors in your breeding program. So if you are sitting in a room and then analyzing data, just not going into the field, not taking a look, then you will mistake them for the real phenotypes, right? So it's really important that these things will invariably be there. Whatever you do, how much ever care you take, they will invariably be there. But then it is important to adjust them. So there is one great way of adjusting those errors. Have you heard about mixed modeling? Mixed model analysis of spatial variation. So you can use mixed modeling techniques to analyze those spatial variations. Now, in fact, I'll show you one picture here. So what you see here is the correlation between two locations. Yield measured in location 4 versus yield measured in location 1 on your left side, right? So what you see is that those dots indicate that there is no big correlation between location 1 and location 4, right? So we think that there is big G by E is playing there. Actually, after you apply the mixed model based spatial correction, what you see here, the same data, after adjusting, you see that there is a clear linear correlation trend between both those locations. See, so that is very important to take out the noise in your data set before you do any downstream analysis. And in fact, in Corteva, this is done very rigorously, in a very sophisticated, using all sophisticated modeling techniques, and it's very much well built into the analysis pipeline. So one is that using those statistical approaches, the other one is using customized machines. So generally what we do is we use whatever machine is which is readily available off the shelf to measure your characteristics. Whereas here, what you see here is called a Boreas wind machine. You know, lodging tolerance in many of the crops is becoming business critical. A plant should not lodge, be it rice, be it corn, be it pearl millet. Many of the crops, lodging tolerance is very, very critical. So how can we develop crops which are lodging tolerant, which will not fall? So one way of measuring lodging tolerance, instead of relying on natural winds and other things, what we have done is there is a machine which is customized for this purpose, and it blows wind at a very high velocity, and it is done on your cornfields as you see, and it will measure, and whatever falls down, that means it doesn't have stock strength. So that's the way it is used in the field. Similarly, in rice, grain quality is one of the most important traits. You know why hybrid rice is not becoming popular is because of the poor grain quality in the past, whereas now those grain quality issues are being addressed, at least in our company, through a visual grain analysis system, wherein each and every grain of rice goes through a 3-dimensional analysis. So you can understand what is the opaque portion, what is the transparent portion, what is the soft endosperm, what is the hard endosperm, and all that information gets used in the selection process. Another very, very interesting development with respect to phenotyping is on drones. How many of you have seen a drone? Many of you have seen. So mostly in marriage parties and big gatherings where they are taking pictures as of today, right? That's quite extensively used, which is a very good application. But then this can be really, really useful in many other fields. Drones, for instance, we use drones in our fields for measuring plant height. When you join a seed industry, the very first job you might be given many of the times would be to go and measure plant height. And it takes, in a large field, it takes almost like 8 to 10 research associates in 2 or 3 acre fields for at least 6 or 7 days continuously you do, then you can measure each and every plant height. Whereas with drones, you set everything, you optimize the protocol, within 15 minutes it just flies over the field and then it gives you back the data. So not even single manpower is used, right? And the time saved. So tremendous application potential for drone use, but then it's also being a regulated space in India. Government regulates use of drones, so I'm sure this will be more unregulated in the coming years, and we are going to see a lot of developments in this space. And another significant thing about phenotyping as well as genotyping is how well you are able to organize the information. That's what I told you earlier. We are generating millions and millions of data points, but how to make sense of those data points in the breeding program so that the breeder can select meaningfully on those data generated. So that is the real challenge currently. So you need to have good clusters, performance clusters, and then you need to have highly information in a really a manner in which you can really easily visualize. So all these graphics are a must, and high performance clusters are must, and this is where a number of breeding programs which are aspiring to be modern are spending resources currently. Okay, and one important development in this field is cloud-based applications. I've heard about cloud-based computing, cloud-based storing of the data, AWS, Amazon Web Services, right? So a lot of development is happening there. Even companies or organizations which do not have significant resources to invest in this field can just make use of cloud-based systems, so that you don't need all these hardwares that picture says. No, they may be millions of costing dollars, whereas all this can be done now in cloud. So if you ask me, are there examples of using all these technology and developing a product? So how many of you have heard about a product called Aquamax? So Aquamax is a drought tolerant, water use efficient product released from Corteva, which has become very, very popular in North America. So currently we are also in Asia, we are working on this Aquamax technology. So Aquamax is nothing but whole genome prediction was used, the genomic selection was used, managed stress screening was used, which is a biotic stress measurement technique, and many of the precision phenotyping techniques were used, and that resulted in water use efficient hybrid. So there are practical examples that all these technologies can be integrated into your product pipeline. So these are not anymore just theoretical or potential applications, these are actually realized applications in the field. So three paradigm shifts we have seen. The first one is line development, genotypes, phenotypes, and the fourth one is the breeding approach itself. We have been for long focusing mostly on the quantity, yield. We were mostly focusing on yield. Whereas of late we have started thinking about quality of the produce. So can we develop products that are nutritionally rich in value? So what are we doing in this space? What are the breeding programs doing in this space? So one of the things that the Corteva has done, the company, it's called Plenish soybean. So this soybean it's a genetically engineered soybean mostly in North America and other parts of North American continent. So here what has happened is the proportion between saturated and unsaturated fatty acid, right? So that is deliberately shifted in favor of unsaturated fatty acid, and the saturated fatty acid is actually less. So this is a very healthy product, and this is now one of the fast selling products in North America. So it is possible to develop high quality products using these techniques. How many of you have heard about quality protein maize? Quality protein maize that is rich in lysine and tryptophan, and it has double the amount of lysine and tryptophan. I had the privilege to work on quality protein maize for quite some time when I was with ICAR and later with CIMMYT. So in fact we developed, when I was working for ICAR, we developed the very first marker assisted selection product called Vivek UPM9 that had almost twice the amount of lysine and tryptophan as compared to the normal maize. And this area is also becoming very, very popular. But the only issue here is whether whatever products we are generating, value added products, are they getting premium price in the market? So what's happening is the farmer who is producing these premium products, he is not getting the premium price. So that's why these products are not fast moving, but then all this scientific success can indeed translate into human benefits. Maybe not today, but maybe 5-10 years from now, it can become potentially. Similarly, pro-vitamin A hybrids. So TNAU has been working, Dr. Sendil has been working on this, and I think we are very close to releasing hybrids with this trait. Beta carotene trait, very vital for immunity, for eyesight, a number of health benefits. Corn, which has 14 to 15 times higher beta carotene as compared to normal corn. So now this is also possible through molecular breeding approaches. So that was the fourth paradigm shift, that it is not just the quantity, quality is also equally important. And the fifth one that I want to talk about is the onset of disruptive technologies, right? So I'm sure you all must be hearing a lot, reading a lot about disruptive innovations, innovations that have tremendous impact and that just changes the way you do everything. There are so many examples of disruptive innovation outside agriculture, right? All the internet banking or UPI interface and all the digital technologies that we are using every day in and day out, They are all really, really powerful technologies, and they have changed the way e-commerce, right? The way you are ordering groceries, you are buying your own clothings, everything now. So that has completely changed the way we do shopping. Similarly, in agriculture also, there are a lot of things that are changing. So one of the disruptive technology, anybody can tell me one example of a disruptive technology? Biotech students? Anything that you think can be disruptive? Heard about genome editing? CRISPR-Cas, right? So this technology has the potential to change the way we breed. So there is a lot of potential here. So Corteva has a state-of-the-art actually genome editing center based out of Johnston in Iowa. So in fact, we developed the very first product that has been commercially approved for cultivation in the US, and it's called Waxicon. A very niche segment, very small segment, only it has market. But then it was scientifically proved that yes, such technologies can be indeed developed and it can result in a commercial product. So this technology has a lot of potential, but the only issue is, again, comes back to our breeder's interest. Most of the breeder interested traits are complex. So can editing one or two, will that be helpful? So we need to think about genome-wide editing and genome-wide computational parameters, how we can do that. And another important technique is reverse breeding. I don't know if you have heard about it, just go back and read about this. Generally, you know, if you have a hybrid, generally if you have a parent, you can form a hybrid. But you cannot recover, if I give you an F1 hybrid, you will not be able to recover the inbred lines from that hybrid, right? Currently, will not be able to. But then this conceptual paper initially and then later experimentally demonstrated also that it did prove that from an F1 hybrid, you can in fact isolate the parental lines. So you can suppress recombination in such a way that the parental combinations can indeed be identified, and it was experimentally, empirically demonstrated, and it was very well published in Nature, Science, and all those magazines. Another important thing, you know, when I read about this future food thing, I was really alarmed. Why? As a corn breeder, I was alarmed. Why? Because most of the corn that we produce goes to as poultry feed, right? So what these guys are saying, a bunch of new smart youngsters like you guys, okay? So what these guys are saying is that if I have to eat chicken leg, I don't have to grow the chicken. I can just grow the chicken leg. I can just grow the chicken breast. I don't need to. So this was indeed actually in 1932, Winston Churchill, he did make this statement that it is very absurd to be able to eat a small part of chicken, you need to grow the entire chicken. So that guy was so visionary in 1932, but that is coming true now. It is possible that through cell culture, through tissue engineering, we can grow parts in labs, only the parts that we want to grow, so that you are not creating a life, but then you are creating life parts. You know, science is progressing so fast that these things are indeed possible. In fact, the very first burger was actually produced through this method, and you know how much it costed? $330,000. $330,000 for one piece of burger, right? But then the science and technology also improved so fast that within a period of 2 or 3 years, they were able to produce a burger, a company called Mosa Meat. From $330,000, they came down to $30. So all the ingredients required for a chicken leg is put in a culture plate, and then you grow the meat, so that you don't have the taboo of eating non-vegetarian food. So things are evolving fast, but then the basic breeding approaches, the basic genetic approaches remain the same. Maybe now we are breeding at a plant level, maybe then we will be breeding at a cell level. So as long as you are good with breeding, you can still manage this technology change also, okay? So that's my last slide. I just want to leave you with this impression that a lot of changes have happened, paradigm shifts have happened in plant breeding. So what has it resulted in? In the past, you know, how many of you have heard or read Rex Bernardo's paper? Rex Bernardo is a professor of plant breeding at University of Minnesota. He writes really good papers. It's very easy to read, very easy to understand. If, as a plant breeding student, if you have not read Rex Bernardo, please go back and read. He has published lots of papers. So he describes in one of his books plant breeding in just one sentence. So he says plant breeding is nothing that you select the best, two very good plants, make crosses, cross the best, and then you hope for the best. That's what practically, I mean, I'm not taking away any credit from the plant breeders. That's what we generally do. We take good by good, we make good by good crosses, and then from the progenies we select something which is better than the parent. And then if you ask for population size and so many other details, every breeder will have his own population size, his own philosophy of breeding, but then everybody hopes that something better will come out of my cross. Okay, so that's how plant breeding has operated until now. But what's happening with all the technologies, not just molecular breeding or line development or precision phenotyping, breeding informatics, all these package of technologies, what has it done is now the paradigm has shifted towards this. It remains the same, select the best, you are crossing the best, but the rest of the things you are not leaving it to hope, you are moving in a clear direction with assured confidence and guaranteed success. That yes, this is what I want, and I can assemble my genotype in such a way that it will give me a desired phenotype. So that the ability to move towards a clear defined target phenotype through manipulation of genotype has become an important component in the current modern day breeding practice. So with that, I would like to close this, and I would like to thank all my colleagues from Corteva who have contributed to this presentation, and also my ex-colleagues, CIMMIT as well as ICR, where I spent quite a bit of time. Thank you very much once again.